How does an enzyme speed up a reaction? Enzyme greatly increases the local concentration of the reactants at the catalytic site, holding them in the correct orientation for the reaction that is to follow. So basically an enzyme is going to bring the reactants near each other and thus increasing the local concentration of them. And that leads to speeding up the reaction. Also, holding them in the correct orientation helps so that the reactants can attack each other and form these new covalent bonds more easily. Enzymes have a much higher affinity for the transition state of the substrate than they do for the reactants, both reactant and product. Thus, if we have E plus S going to ES, that binding reaction has a delta G standard for ES forming, which is going to be less than 0. It will favor the complex of ES. E plus the transition state will also favor the complex of enzyme and transition state with a negative delta G standard. E plus product going to E product will also favor the complex of enzyme product. But the relationship is that the enzyme is going to hold on much more tightly to the transition state than it will to either one of the other ones. Why? Well, the transition state is a transitory molecule that in the active site, it will be held there to either turn into product or be turned back into substrate. So the relationship of the delta G standards will look something like this, where the delta G standard for the transition state with the enzyme is going to be lower than either one of the other two. Graphically, it will look something like this. We have the normal reaction without the enzyme in red. So that's A right here. And we have the one with the enzyme involved, being catalyzed. And the energy of activation for the uncatalyzed reaction is being shown in red arrow right here. And the energy of activation for the catalyzed reaction is being shown in green. And of course, the EA for the catalyzed reaction is much smaller than the EA for the uncatalyzed reaction. Now, binding of the enzyme to the substrate has a negative delta G standard, so that actually brings the ES lower than the substrate. So that's what's happening right here. And enzyme binding product stabilizes the product because it has a negative delta G standard, so that brings the enzyme product down, and that's what you can see. The enzyme product is a little bit lower than the product. But it's that affinity that the enzyme has for the transition state being shown here that lowers the transition state to a much lower level compared to the uncatalyzed reaction. So you can see that this difference, some of this difference in the EA, or the state of where you have the transition state up here going down to here, some of that is due to the negative delta G standard for binding of the enzyme to the transition state, where it's holding tightly to that transition state. Now, once the transition state is turned into product, now the enzyme can more easily let it go because the binding is not as strong for the product or the substrate. So you don't necessarily want the product to be held on too tightly by the enzyme, neither the substrate too tightly by the enzyme. And to show this, we're going to go ahead and take a look at it in a little bit more detail. So here we have an uncatalyzed reaction of substrate going to product, okay? And we have an EA for this, which is being shown right here. Now, when we are looking at the reaction being catalyzed, and this is the pathway being shown right here for the catalyzed reaction, we're going to go step by step. So first step is enzyme binding substrate, this first part right here, right? So we have an ES, and we're going to put the ES because the formation of ES is favorable, or it has a negative delta G standard, so it's going to be somewhere down here. So initially, you have the binding of enzyme to substrate, and you go down here. Now the next part is ES to EP, but we know that this part right here, there's a transition state in the middle right here. You're not going to go straight from substrate to product. So you have to go through the transition state, and let's put the transition state for the uncatalyzed reaction right here, ST, and we're going to put the transition state here, EST. So the reaction moves up to there, but since the enzyme is binding tightly to the transition state, part of this difference between here and here is that negative delta G standard of binding for the enzyme to the transition state. So now the transition state will be transformed to the product, and we have enzyme product which will be at a lower level than the product itself because the enzyme binding the product will have a negative delta G standard. So we're going to go down here and bind there, and then the product will be released and you go back here. Okay, so that's what it looks like. Now, if the enzyme was binding to the substrate much more strongly, what will happen is, let's have a mutated enzyme, and we'll just put a star on it for being a mutated enzyme, but it binds a substrate much more strongly. So you're going to be down here somewhere. And now it's going to go back up to here, and then down here, down here. Our mutation does not affect how strongly the enzyme binds the transition state or how strongly the enzyme binds the product. So let's look at the energy of activation for these reactions. So the first one right here, the difference would be right here between these two states. So this is the unmutated enzyme. And we can see that the EA is smaller than the non-catalyzed reaction. Now for this mutated enzyme that binds a substrate much more strongly, the difference is this. Right? You can see the difference is much bigger. The energy of activation is much higher. It's even in this picture that we have, the energy of activation of this mutated enzyme catalyzed reaction is higher than the uncatalyzed reaction. So an enzyme binding its substrate very strongly is not actually a good thing. If we had another version of this mutated enzyme where it bound the product more strongly, you run into another issue because then the product won't be released as easily, and you'll have a bunch of enzyme with the product still in the active site not being released, so substrate can't get in there for further catalyzation. Note that increasing the binding strength of the enzyme for its substrate while not affecting the binding strength of the enzyme for the transition state can increase the EA, therefore decreasing enzyme efficiency. And that's what we talked about. Okay, so moving on from enzymes to the cell membrane, and there's just some basic information about the cell membrane because this is going to be important to what we'll be talking about in section 2. A cell's membrane contains lipids and proteins. And this is just a cartoon of the cell membrane being shown here. One of the primary lipids in all biological membranes are phospholipids. A phospholipid has a hydrophilic head and a hydrophobic tail. And here is phosphatidylcholine, one type of phospholipid. And you can see here that you have the fatty acid tails right here, 2 of them. You have a glycerol right here, a phosphate that is going to be negatively charged right here, and then you're going to have a different type of head group depending on the type of phospholipid you're dealing with. So in this case, for phosphatidylcholine, you have a choline group. Now, the choline group has one positive charge on it, as being shown here, and the phosphate has one negative charge on it, being shown here. So overall, we say that this phospholipid is neutral. It doesn't mean that that positive and negative charge don't exist. They definitely exist. And that is a big part of why the head group is hydrophilic, right? But overall, when we're looking at the molecule, we can say, well, there's one negative charge and there's one positive charge. +1 plus -1 equals to 0 in the case of phosphatidylcholine. Different phospholipids have different head groups, but are structurally similar. So in this case, you can see that we have a picture of 4 different phospholipids: phosphatidylethylamine, phosphatidylserine, phosphatidylcholine, and sphingomyelin. Now, these all have very similar structures. You have phosphate, 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 right? And then you have 2 fatty acid tails, or fatty chains, all here, right? 2 here, 2 here, 2 here, 2 here. Now, the head groups are different. So you can see that the head group of this one is different than the head group of this, then it's different than this. Now, these 2 right here, so let me erase this box right here. These 2 right here are the same, but sphingomyelin is different than the other phospholipids. So phosphatidyls have a glycerol. So you can see that here's the glycerol group right here. Here's another glycerol group right here. Here's another glycerol group right here. But sphingomyelin comes from sphingosine right here. And it's a little bit different because sphingosine has 1 fatty chain, and then you attach another fatty chain right here. And that's where that NH group you can see goes from NH3 plus to NH. And then you add the phosphate to this head right here. Okay? So even though they have the same head group, which is the choline group, the structure of a sphingomyelin is different, the base structure. It doesn't come from a glycerol being attached to 2 fatty acids, basically. So that's the difference. Now, getting rid of all this arrows and stuff here. So, if we look at these, you can see that in the case of phosphatidylethylamine, you have 1 negative charge and 1 positive charge. So this is going to be overall neutral. And that's what's being shown in this cartoon drawing right here. We know that there's a full negative charge and a full positive charge there, but overall the whole molecule is neutral because those 2 things cancel out each other. Doesn't mean they disappear, they just cancel out each other when we're looking at the whole molecule. In the case of phosphatidylcholine, it's the same thing. 1 negative, 1 positive. And again, you have overall a neutral phospholipid. Sphingomyelin is the same thing. 1 negative, 1 positive. And they cancel out each other for being a neutral phospholipid. Now, in the case of phosphatidylserine, you have 1 positive and 2 negatives. And this gives phosphatidylserine an overall negative charge on it. Now, this is important because phosphatidylserine is special, and we'll get into some of the reason why it's special a little bit later. Now, there's another type of lipid in cell membranes, glycolipids. Glycolipids are components of a cell's membrane. They're similar to phospholipids, but have sugars as the hydrophilic head instead. So you can see here that we have basically the similar structure starting out with this hydrophobic area right here, right? Hydrophobic right here, the fatty acid tails or the fatty chains. There's hydrocarbons. But instead of having that choline group and phosphate or some sort of phosphate and charge group or etc. or head group, what you have is sugars. Here's the sugars. Here's a bunch of sugars together. And sugars are hydrophilic. You know that, right? Because you put sugar in your drinks, to your coffee, to your tea, sugars into sodas and etc. And they go into solution very well, so they're hydrophilic. So that creates a hydrophilic head and a hydrophobic tail. Very similar structure to phospholipids. Glycolipids like phosphatidylserines are a little bit special, and we're going to talk about that soon. Cholesterol is also a component of biological membranes. And here is a picture of cholesterol. And cholesterol, this whole part right here, is hydrophobic, but this little bitty OH group is hydrophilic. So this little bitty OH group is going to be sticking out into solution while the rest of this is buried in the phospholipid bilayer. Formation of a bilayer is a spontaneous process. So when we say spontaneous, we mean the process has a delta G that is negative. It will happen by itself. No need for an input of energy or coupling this to any other type of reaction that is favorable. So we have two types of shapes for molecules that are amphiphilic. And remember what we said amphiphilic is? Amphiphilic is any molecule that half of it is hydrophilic and the other half is hydrophobic, right? So if the molecule kind of looks like a cone, like right here, what will happen is if you throw it in water, it will form a micelle. Now, why will it form a micelle? Well, the fatty acid tails, or the hydrophobic part, is hydrophobic, right? And what will water do when it comes near a hydrophobic chain or hydrophobic surface? It will try to minimize its interaction with that surface. The only way to minimize that interaction to zero is by putting all those chains right here into the core of the micelle, away from the aqueous solution, away from water, so water doesn't form those ice-like cages around it to create more order. So they're away from water, so no ice-like cages, and all the hydrophilic heads are now facing solution. They're hydrophilic. They can interact easily with water, right? Now, when you have a molecule like our phospholipids, well, they're not going to be cone-shaped. They're going to be cylindrical shaped like this. And the only way that you can minimize the interaction of water with the hydrophobic tails is that you form a lipid bilayer. Now you can see here that all the lipid tails are in the core, right? Away from solution. So water is minimizing its interaction with those tails by shoving them into the middle of the lipid bilayer. And all the heads that are hydrophilic are facing solution, happily interacting, forming hydrogen bonds with water, ionic bonds, and etc. So even though there's an appearance of order being created in this case, as far as phospholipid bilayers, for example, the system is becoming more disordered, right? Because having those tails actually interact with water will create order, because water will form those ice-like cages around those hydrophobic surfaces. And to increase entropy, those tails have to be shoved away, excluded from solution, so that water is minimizing its interaction with it, therefore minimizing the amount of ice-like cages formed, or the amount of order formed. But there's a problem, right? So let's go back to this picture. The problem is these surfaces right here, right here. These phospholipid tails are still facing the solution. And how do you get rid of that? Well, once you have the bilayer and you have those exposed edges fold on each other to create a sphere being shown here. Now, none of the hydrophobic surfaces are exposed to solution, so the solution has minimized, the aqueous solution has minimized its interaction with the fatty acid tails, with the hydrophobic surfaces, to zero. So the result is that you have two compartments. Here are my hydrophilic heads. I'm going to show them with two circles. And in between you have the fatty acid tails, which are hydrophobic. And what you have now are two aqueous solutions. So 1 and 2 that are separated from each other by this phospholipid bilayer. This is a way of creating two compartments and it happens spontaneously. If I had some phospholipids, which you can buy from a vendor, you buy them, they come in a sort of a powder format, and you go ahead and put them in water, those phospholipids will just go ahead and spontaneously start to form these sealed structures. Okay, so movement and distribution of lipids in the cell membrane. Each lipid bilayer consists of two monolayers. For the cell membrane, there is the intracellular inner monolayer of the leaflet and the extracellular external monolayer of the leaflet. Here are my phospholipids forming a bilayer, phospholipid heads that are hydrophilic, and the tail is facing the interior, right? And if this is the extracellular environment, and this is the intracellular environment, the inside of the cell, the cytosol, so the inner monolayer or leaflet would be this, and the extracellular external monolayer or leaflet would be this. Okay? Amphiphilic molecules such as phospholipids and glycolipids can freely diffuse within the leaflet they are on, rotate around their own axis, but cannot flip from one leaflet to another without the help of an active process within the cell. So we have a picture here that shows this very nicely. A phospholipid, let's say this one right here, I'm just going to color the head green, can move on the leaflet it is on, freely diffuse in any direction it wants to, this way or this way. It can sort of float around on that leaflet. It can rotate around itself, meaning on its own axis just rotate around itself, which is being shown here. But this thing of going from the leaflet it's on and going to this other leaflet, that cannot happen by itself. This is not going to be a spontaneous process. This actually requires some sort of input of energy, meaning coupling the movement of a phospholipid from one leaflet to the next will require coupling that process to a very favorable process, such as ATP hydrolysis or something else. The composition of lipids on individual leaflets are not the same. In mammalian cell membranes, a phosphatidylserine and a phosphatylineacetol is exclusively found on the inner leaflets, whereas glycolipids are found on the external leaflet, phosphatidylcholine is found on both leaflets. This asymmetry is accomplished through active processes in the cell, or in cells. So, here we have organization happening at a different level. Organization that cannot happen spontaneously. So first we have phosphatidylserines and phosphatidylineacetols. These are two types of phospholipids that are only found in a living cell in the inner leaflet. Glycolipids are found only on the external leaflet. Phosphatidylcholine is found on both leaflets. So this picture right here is showing you, so all these phospholipids that have a negative charge, like right here, etc., right? These are all phosphatidylserines because phosphatidylserines are the only phospholipid that overall it has a negative charge. It has one positive and two negatives. You add them up, you get a negative charge. And the glycolipids are being shown right here and here. That's a glycolipid right there. And the glycolipids are on the external leaflet right here. And the phosphatidylserine is going to be on the inner leaflet. Phosphatidylineacetol will also be on the inner leaflet. It's not being shown in this picture, but that's where it's going to be. The asymmetry of the cell membrane is important to biological activity. Phosphatidylineacetol participates in intracellular signaling pathways. This is something that we will be talking about in section 3. Phospholipase C, a cytosolic enzyme, cleaves phosphatidylineacetol into two parts, the hydrophilic head, which diffuses into the cytosol, and diacylglycerol. And diacylglycerol is nothing but glycerol and the two fatty acids. That's all that is. Both of these act as second messengers, so both of these, in single transduction pathways. Again, something that we'll be talking about in section 3, but I have it here as an example to show you why phosphatidylainositol is on the inner leaflet. Glycolipids are used for cell-to-cell recognition. The sugar moieties on glycolipids is different from cell type to cell type. So every different type of cell will have different types of glycolipids on its surface, and that is a way for cells to recognize each other because cells don't have eyes. So they recognize each other by recognizing what's on the external leaflet of that cell. And that is through molecular interactions, binding interactions between, for example, the sugar groups and the glycolipids with a molecule that is on the surface of another cell. Glycolipids also protect the cell membrane in harsh environments, i.e. lung and gut epithelial cells. So cells where their apical surface, and the apical surface in the case of epithelial cells, let me just draw that out. Here's my epithelial cell, right? Here's my basement membrane. The epithelial cells have a basal and apical membrane. The basal membrane is facing the basement membrane, so B to B. So this would be the basal membrane right here. And the opposite of the basal membrane is the apical membrane, and that would be here. Now in the case of epithelial cells, the apical membrane is facing some sort of cavity. So for lung cells, in your airways, the epithelial cells, the apical membrane is facing into the air. So the air you breathe in will come to interact with these apical membranes of the epithelial cells in your lung. Now, that essentially is an outside environment, so it's a harsher environment, and there are lots of glycolipids on these apical membranes that help to protect that apical membrane from that harsh environment. Phosphatidylserine on the external leaflet is a signal to immune cells that the cell is dead, therefore can be phagocytosed. So phosphatidylserines are always found on the inner leaflet. So if they're on the external leaflet, meaning facing the external environment, that means the cell has died. The protein that maintains the asymmetry is called phospholipid translocator. The activity of this protein ensures that phosphatidylserine is exclusively on the inner leaflet. So you have this protein, the phospholipid translocator, that basically makes sure that phosphatidylserine is always on the inner leaflet and not on the external leaflet. During apoptosis, programmed cell death, phospholipid translocator activity is inhibited, and then scramblase, another protein, is activated. Scramblases are proteins that randomly move phospholipids from one leaflet to another. The actions of scramblases has the effect of destroying the asymmetry of phospholipids between the two leaflets. So what scramblase does is it just randomly picks a phospholipid from one leaflet and moves it to the other leaflet. It could be it randomly picks one phospholipid from the external leaflet, moves it to the internal leaflet, or it picks randomly an internal leaflet phospholipid and flips it to the external leaflet. This action is literally like shuffling cards. The phospholipid translocator is putting the cards in order. The scramblase is shuffling the cards. So during programmed cell death, what you have is you shut down the organizer, which is the phospholipid translocator, and you activate the shuffler, scramblases. Because if you have them both active at the same time, you're just wasting energy because the two processes are working against each other. So it's like nothing is getting done. Phosphatidylserine is not present on the external leaflet through the actions of scramblase. Immune cells have cell surface receptors that recognize phosphatidylserine. So a normal cell, a live cell, will not have phosphatidylserine on its surface. So when an immune cell comes near it, that protein that recognizes and binds to phosphatidylserine has nothing to bind to. The binding of phosphatidylserine to these receptors is signaled to the immune cell to phagocytose and therefore remove the dead cell from the environment. So let's take a little bit of a look and just write this out, right? So we have our phospholipid bilayer, and we're going to say this is the external and internal. And we're going to color phosphatidylserine with a little bit of a green. So here's my phosphatidylserine. I have 2 of them here, maybe 3. Let's just go for 3, okay? And we have a process. The cell has gone through apoptosis in one direction. We're going to have this happen, right? We're now, let's say 2 of the green are now on the outside and 1 is on the inside. And this is going to be accomplished by the protein called scramblase. Now, the opposite process of organizing it is done by phospholipid translocators. So translocators. Now, if the cell is alive, apoptosis is not active. Apoptosis is a cellular process that many things happen during apoptosis. So the one mistake that students sometimes make when we're going over this is that they think that the process of scramblase scrambling initiates apoptosis. No. Apoptosis is a multi-step process that happens to a cell. One of the things that happen is this. So if that process of apoptosis has not been initiated, then this part is not active, meaning scramblase is inactive. So this arrow going from organized to unorganized is not working. So the only arrow that's working is the arrow that we have for the translocator, and that is to organize things. Move the phosphatidylserines to the internal leaflet, keep things organized. Now, when apoptosis is activated, then what will happen is scramblase is also activated as part of the process of apoptosis, and the translocators are inactivated. So now that the translocators are inactivated, the only arrow that you have is the arrow of disorganizing and shuffling up things. And then you're going to have phosphatidylserines on the external leaflet. Now, when you have that, you'll have immune cells. Let's scroll down a little bit. What you'll have is immune cells. And here's an immune cell's membrane. And it will have receptors that will bind to and recognize these phospholipids. So I know that I didn't draw out the phospholipid bilayer for the immune cells, but just imagine that that line is the phospholipid bilayer, and this is the membrane of the dead cell, which then goes around like this, right? And this will bind and recognize the phosphatidylserine that is now on the external leaflet, and that will send the signal into the immune cell to go ahead and phagocytose this dead cell and remove it from the environment. Okay guys, we're at the end of this section. I will talk to you guys later. Bye-bye.